Mention whether the following statements are true or false. Mitosis is also known as reductional cell division. False. Plastids and endoplasmic reticulum disappear during the prophase of mitosis and meiosis. False. Karyokinesis refers to the division of the nucleus. True. Before undergoing cell division, the amount of DNA in the cell is reduced to half. False. Crossing over occurs in mitosis. False. In mitosis, prophase is of short duration. True. Histone is a particular type of fatty acid. False. RNA contains thymine. False. Mitosis requires lesser time than meiosis. True. Terminal meiosis is known as gametic meiosis. True. Name the type of cell division that occurs during growth of shoot. Mitosis. Formation of pollen grains. Meiosis. Repair of worn out tissues. Mitosis. এবার মাইটোসিস আর মিওসিসের ডিফারেন্স Mitosis, it is an equational division. Meiosis, it is a reductional division. Mitosis, two diploid daughter cells are formed. Meiosis, four haploid daughter cells are formed. Mitosis, only one division occurs. Meiosis, two divisions occur. Mitosis, it occurs in somatic cells. Meiosis, it occurs in germ cells. Next question, centrosome and centromere difference. Centrosome, it is an organelle of the animal cell. Centromere, it is a non-stainable part of chromosomes at which two chromatids join. Non-stainable part of chromosomes at which two chromatids join. এবার centrosome, it contains two centrioles which move towards the opposite poles and form spindle fibers during cell division. Centromere, it provides site for attachment of spindle fibers during cell division. Next question, cytokinesis. It is the division of cytoplasm. Karyokinesis, it is the division of nucleus. Cytokinesis, it starts after karyokinesis. Karyokinesis, it is the first division. Chiasmata. It is the point of attachment of non-sister chromatids of homologous chromosomes where crossing over takes place. Ever crossing over, it is the exchange of genetic material between two sister chromatids of homologous chromosomes. Next, G1 phase and G2 phase difference. G1 phase, it is called first gap phase. G2 phase, it is the second gap phase for growth phase. G1 phase, no change occurs in its DNA content. G2 phase, amount of DNA is double than G2 phase. G1 phase, it is close to the S phase or synthetic phase. G2 phase, this phase is close to M phase. G1 phase, it is the most variable with regards to duration. It either occupies 30 to 50% of the cell cycle or lacks entirely. G2 phase, it may occupy 10% to 20% of the cell cycle. Next, what are centrosomes? Centrosomes are highly condensed chromatin fibers. In 1875, it was discovered, this thread, E. Strasburger discovered this thread-like structure during cell division. What is centrosome? It is a small hyaline body situated just near the nucleus in majority of the cells. It consists of two minute dot-like structures called the centrioles. What are the acrocentric, submetacentric, and metacentric chromosomes? According to the position of centromere, chromosomes are classified as acrocentric, submetacentric, metacentric, acrocentric, having terminal centromere, submetacentric, having subterminal centromere, metacentric, having median centromere. What is satellite chromosome? Chromosome having a satellite serves as a marker chromosome called satellite chromosome or sat chromosome. What are telomeres? The chromosome extremites or terminal region on either side. The chromosome extremities or the terminal region on either side of the chromosomes are called telomeres. Chromosome extremities or terminal region on either side of the chromosomes is called telomeres. Define karyotype and ideogram. Karyotype. The term karyotype is given to the group of characteristics that identified as a particular set of chromosomes in individuals of the same species. Ideogram. When a particular set of chromosomes after identification by karyotype is represented by a diagram, it is called ideogram. When a particular set of chromosomes after identification by karyotype is represented by a diagram is called ideogram. What is chromatin? When interphase nuclei is dipped in 0.5 m solution of NaCl, it shows a network of long, extremely thin, dark staining fibers. These are chromatin fibers. These are located in the nucleus of the cell. What are the constituents of a chromatin fiber? Chromatin material that constitutes the fiber is formed of two substances, DNA and histones. DNA is deoxyribonucleic acid আর histones particular type of basic protein. DNA হচ্ছে 40% থাকে আর histone 60%. What are euchromatic and heterochromatic chromatin? Euchromatin and heterochromatin. Euchromatin, when chromosomes are stained with acetocarmine or fulgen. Fulgen. The part of the chromosome which is highly stained is called euchromatin. It is genetically active and considered to be partially or fully uncoiled. Heterochromatin, darkly stained region of chromosome is called heterochromatin. It is generally inactive and tightly coiled part of chromatin fiber. What are chromocenters? Chromocenters are heterochromatic regions of varying size which occur near the centromeres in proximal region of chromosome arms. What are constitutive and facultative heterochromatin? Constitutive heterochromatin. Certain region of chromosome proximal to centromere is constant and serve as a marker called constitutive heterochromatin. Facultative heterochromatin. Other region of chromosome beside constitutive part is called facultative heterochromatin, such as whole sex chromosome becomes heterochromatic only at a certain stage. What are nucleosomes? Nucleosomes are the complexes in which DNA wraps around a protein core of 8 histone molecules. These are 10 nanometer in diameter. What are histones? Histones are the basic proteins around which DNA winds. DNA mane 200 BP winds. How many types of histones are there? There are 5 types of histones: H1, H2A, H2B, H3 and H4, where H2A, H2B form a heterodimer. H3A, H4 form a heterodimer, and altogether H2A, H2B and H3, H4 form a tetramer. H1 clips the DNA around the protein core. Describe the structure of DNA. The two strands of DNA are anti-parallel to each other and are made up of small subunits called nucleotides. It is also a macromolecule. What is the composition of DNA? DNA is composed of repetition of nucleotides, which is shown in the following schematic view. ডিএনএ থেকে নিউক্লিওটাইড, নিউক্লিওটাইড থেকে নিউক্লিওসাইডস অ্যান্ড ফসফরিক অ্যাসিড, নিউক্লিওসাইডস থেকে পেন্টোজ সুগার আর নাইট্রোজিনিয়াস বেস, পেন্টোজ সুগার থেকে ডিঅক্সি রাইবোজ অ্যান্ড রাইবোজ, আর নাইট্রোজিনিয়াস বেস থেকে পাইরিমাইড আর পিউরিন। এবার পাইরিমাইড থেকে TCU আর পিউরিন থেকে AG। Next, define nucleosides and nucleotides. Nucleosides are those molecules whose nitrogenous base is combined with sugar molecule. In DNA, four different nucleosides are formed: deoxycytosine, deoxythymide, deoxa or deoxyadenosine, deoxyguanosine. About nucleotides are derived from a nucleoside by the addition of a phosphoric acid molecule. The phosphate molecule is linked with sugar molecule at carbon number 5 or at carbon number 3. Four nucleotides found in DNA are deoxycytidyllic acid or deoxycytidylate, deoxythymidyllic acid or deoxythymidylate, deoxy adenylic acid or deoxyadenylate, deoxyguanylate acid or deoxyguanylate. What is replication? Replication is the process by means of which DNA is synthesized by copying of complementary strand of duplex DNA. Replication is a process by means of which DNA is synthesized by copying of complementary strand of duplex DNA. What are the modes of replication? Replication occurs in three manners by modes: conservative, semi-conservative, dispersive. Conservative. In conservative replication, double stranded molecule is conserved as such and that a new copy is synthesized from the old molecule. Semi conservative. In this mode of replication, new copy is synthesized by taking each of the parental duplexes. Next, dispersive. Dispersive key. In this mode of replication, old molecules should disintegrate and two new molecules should be synthesized. What are genes? Genes are specific sequences of nucleotides in a chromosome that encode particular proteins which expresses in the form of some particular features of the body. What do you mean by cell cycle? Every cell capable of cell division passes through different stages or phases in a cyclic manner. It is called cell cycle. What type of cell division occurs in somatic cell of the body? Mitotic cell division. Where does the meiosis occur in our body? In reproductive cells. What is the importance of meiosis? It maintains the constant number of chromosomes in a species. It also brings about variations which result in the evolution or origin of new species. Why is meiosis referred to as reductional division? It is referred to as reductional division because the number of chromosomes in daughter cells is half than that of the mother cell. Next question: what is interphase? Interphase is the long resting phase in the cell cycle during which cell grows and multiplication of DNA, synthesis of RNA and proteins take place. Differentiate between karyokinesis and cytokinesis. Karyokinesis is the division of nucleus and cytokinesis is the division of cytoplasm. State the specific term for each of the following: nuclear division, karyokinesis. Point at which two sister chromatids are held together, centromere. Phase in the cell cycle where protein and RNA are synthesized, G2 phase. The period between the two successive mitotic divisions, interphase. What Next question: what are the four essential stages that take place during meiosis? The successive division without DNA replications occurring between them, the formation of chiasmata and crossing over for genetic variability, the segregation of homologous chromosomes in meiosis, the separation of sister chromatids in meiosis. What is the full form of RNA and DNA? RNA ribonucleic acid, DNA deoxyribonucleic acid. What is nucleoid? Nucleoid. Nuclear material of a prokaryotic cell is called nucleoid. What are chromosomes made of? Chromosomes are made up of DNA and histone proteins. Where are genes located? Genes are located in the chromosomes. What are the major activities of mitosis? Duplication of DNA, karyokinesis and cytokinesis. Name the phases of cell cycle: G1, S phase, G2 phase and M phase. Which structure of animal cell forms the aster of spindle? Centrosome. What is the other name of interphase? Resting stage. Name the chemical that arrests cell division in the cells. Colchicine. Colchicine. Name the chemical that arrests cell division in cells. Colchicine. How do the chromosomes look like during um, metaphase stage of mitosis? During metaphase stage of cell division, the chromosomes look like the shape of a rod, and each chromosome consists of two parallel strands called chromatids, which are attached at a point called primary constriction or centromere. What is the function of chromatin material? The chromatin material mainly consists of DNA, which is responsible for the storage and transmitting the hereditary characters from one generation to another. Why is cell division necessary? Cell division is essential for growth. Every organism begins its life as a single cell. This cell divides repeatedly to produce a cluster of cells. These cells form tissue and then organs to form different activities of life. What are homologous chromosomes? The chromosome pairs which are exactly singular and bear genes at same loci are called homologous chromosomes. What are the major activities of mitosis? The major activities of mitosis are duplication of DNA, karyokinesis, cytokinesis. What is genome? It is the complete set of DNA instructions or single set of chromosomes or genes in a cell. Complete set of DNA instructions or single set of chromosomes or genes in a cell. What is kinetochore? The part of chromosome for the attachment of chromosomal fibers is called kinetochore. What is meant by centromere? What is its significance? Centromere is a point where two chromatids of centrosome a chromosome are attached. It provides site for the attachment of spindle fibers during cell division. It determines the shape of chromosome depending on the position of centromere. A centrosome may have either equal arms or one long and one short arm. Name the cell division concerned with cancer. Mitosis. এবার একটা ছবি আছে সেটাতে তার নিচে কোয়েশ্চেন নেম দ্য টাইপ অফ সেল ডিভিশন মাইটোসিস ডিফাইন দ্য টাইপ অফ সেল ডিভিশন process of cell division where chromosome number remains the same is called mitosis. What is the significance of the above cell division? The chromosome number remains the same both in mother cell as well as in daughter cell. Where does the above cell division take place? The cell division takes place in body cells resulting in growth and development. Number 5, how many chromosomes are present in the above diagram? 4 chromosomes. Next is it a plant cell or animal cell? Give reason. Plant cell because centrosomes with centrioles are absent in the above diagram as it is a plant cell division. Meiosis. Name the type of cell division found in reproductive cells. Is it a plant cell or animal cell? Give reason. It is a plant cell because centrosome with centrioles are absent in the above diagram, so it is a plant cell division. Next, name the type of cell division found in reproductive cell. Meiosis. Next, draw the types of chromosomes according to the position of centromere. Position of centromere অনুযায়ী ক্রোমোজোমের ড্র করতে হবে অ্যাক্রোসেন্ট্রিক টেলোসেন্ট্রিক সাবমেটাসেন্ট্রিক মেটাসেন্ট্রিক acrocentric rod shaped with centromere at one end dividing into into a very short and long arms. Telocentric, rod-shaped, having centromere at the proximal ends. Submetacentric, J-shaped or L-shaped, having centromere almost at the middle position. Metacentric, V-shaped, having the centromere at the center and forming two equal arms. Next, below is the picture of a giant chromosome. See the picture and answer the following questions. Name the centrosome. Chromosome puff. Na. Centrosome. It is a polytene chromosome. Name the chromosome. It is a polytene chromosome. Mark the parts 1 to 4. Chromosome puff, chromonemata, dark band, interband. Where can you find it? The chromosome is found in a dipteran insect Chironomus. Chromosome is found in a dipteran insect Chironomus. Who discovered this type of chromosome? Polytene chromosome was discovered by Indian scientist Balbiani. Draw a neat label diagram to show the metaphase stage of mitosis in an animal cell having 6 chromosomes. একটা বল এঁকে দিয়েছে গোল দিয়ে আর ওই সেই মরণ কুঁয়োর মতো ডট 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 দিয়ে জুড়ে দিয়েছে, দিয়ে লিখেছে ডুপ্লিকেট ক্রোমোজোম, ওই চুলের ঝুঁটি বাঁধাগুলোকে দেখিয়ে, দুটো গোল্লাকে দেখিয়ে, দুদিকের দুটো গোল্লা যেখানটায় বেলুন বাঁধা থাকে সেই বেলুন বাঁধা মতো জায়গাটাকে লিখেছে অ্যাস্টার্স, আর গোলটাকে বলছে সেন্ট্রিওল, ওই বাঁধা গোলটা, আর ওই যে ডট 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 দাগগুলো ওগুলোকে বলছে স্পিন্ডেল ফাইবার্স, আর ঝুঁটির মতো গুলোকে একবার বললো ডুপ্লিকেট ক্রোমোজোম, আর একটা ঝুঁটিকে দেখিয়ে বলছে ক্রোম্যাটিড, জানি না। At the end of mitosis, two daughter cells are formed, and at the end of meiosis, four daughter cells are found. Chromatid, duplicate chromosome consists of two identical strands. Each of this is called chromatid. Centromere, it is the point at which two chromatids join together. Haploid, it is the condition in which a cell or nucleus bears or has a single or unpaired set of chromosomes. The figure below shows a certain stage of mitosis. See the figure and answer the following questions. ওই ঝুঁটির মতোটা হচ্ছে মেটাফেজ, ক্রোম্যাটিড, সেন্ট্রোমিয়ার, সেন্ট্রিওল, স্পিন্ডেল ফাইবার, চারটে হচ্ছে লেবেলিং। ঝুঁটির মতো একটা ক্রোম্যাটিড, ঝুঁটির যেটা মাঝখান সেটা বলছে সেন্ট্রোমিয়ার, সেন্ট্রিওল হচ্ছে ওই মাঝখানটা পুরো, মানে একটা গোলকে হাফ করলে যেইখানটা হয় সেটা হচ্ছে সেন্ট্রোমিয়ার, মাঝখানটা ইকুয়েটারের মতো যেটা মাঝে। সেন্ট্রো না সেন্ট্রিওল, ঝুঁটির ইয়েগুলো হচ্ছে ক্রোম্যাটিড, মাঝখানটা ঝুঁটি হচ্ছে সেন্ট্রোমিয়ার, আর সেন্ট্রিওল হলো ওই মাঝের ইকুয়েটারের মতোটা, আর 4 নম্বরটা হচ্ছে ওই ডট 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 ডটটা হচ্ছে স্পিন্ডল ফাইবার। Three chromosomes are shown in the diagram. How many chromosomes are shown in the diagram? 3 chromosomes. The diagram below represents a stage of cell. Name the stage. Anaphase. It is an animal cell or plant. Is it an animal cell or plant cell? It is a plant cell because centrosome and centrioles are absent. Name the stage that comes prior to this and the stage that comes next to it. The stage prior to it is metaphase and next to it is telophase. Next chapter, absorption by roots. Water is a vital necessity for all plants. It is necessary for hydration of the protoplasm, which in turn is absolutely necessary for proper organization and correct functioning of protoplasm. Next, terrestrial plants are dependent upon soil for the supply of water. Number three, absorption of water by plants is affected by three processes: diffusion, osmosis, and imbibition. Number four, diffusion is defined as the movement of elements or molecules of a substance from a place of higher concentration to a place of lower concentration. Number five, the entry and exit of gases like carbon dioxide and oxygen is the process like photosynthesis and respiration, as well as the movement of gases in the intercellular spaces take place by process of diffusion. Number 6, rate of diffusion is governed by the number of particles per unit volume, density of medium, distance through which diffusion is to occur, are temperature and pressure. Number 7, osmosis is a special type of diffusion that occurs through a semi permeable membrane. Number 8, depending upon the permeability of solvent and solutes, three types of membranes have been recognized. These are impermeable, permeable, and semi permeable. Number 9, impermeable membrane does not allow either the solvent or solute to pass through it. Permeable membrane allows both solvent and solute to pass through it. A semi-permeable membrane does not permit the movement of solute particles across it. Cell membranes are selectively or differentially permeable. They are normally semi-permeable but allow selective passages of solutes through them. Osmosis is the process of passive diffusion of water or solvent across a semi-permeable membrane from the side of low solute concentration to the side of high solute concentration. Osmosis is the movement of water from higher chemical potential to its lower chemical potential without allowing the diffusion of solute by means of a semi-permeable membrane. A solution which can cause osmotic entry of water into it is said to be osmotically active solution. Next, the degree of osmosis depends upon the strength of the solution or osmotic concentration of the solution. Tonicity. Next, tonicity is the measure of osmotic pressure of two solutions separated by a semi-permeable membrane. In relation to cell sap, solutions can be of following three types: isotonic, hypotonic, hypertonic. Osmotic concentration of a solution which is similar to that of another solution, isotonic. Hypotonic, osmotic concentration of a solution which is less than that of another solution. Hypertonic, osmotic concentration of a solution which is higher than that of another solution. Next, osmosis is of two types: exosmosis and endosmosis. Endosmosis is the osmotic entry of water or solvent into a cell, organ, or system. Exosmosis is the osmotic withdrawal of water or solvent from a cell, organ, or system. Osmotic pressure is the maximum pressure which can develop in an osmotically active solution due to entry of solvent into it under ideal conditions when the solution is separated from its pure solvent by a semi permeable membrane. Next, turgidity is the condition of cells, tissues, or organs where they are distended due to osmotic entry of water in them. Flaccidity. Flaccidity is the condition by which cells, tissues, or organs appear limp and flabby due to the loss of water. Targar pressure. Targar pressure is the osmotic entry of water in cell, organ, or system which results in the development of pressure in the confined solution. In other words, Targar pressure is the pressure which develops in the confined part of an osmotic system due to osmotic entry of water into it. It is called hydrostatic pressure or pressure potential. Due to Targar pressure, the protoplast of a plant cell will press the cell wall to outside. Next, the cell wall during elastic presses cell wall being elastic presses the protoplast with an equal and opposite force. This force exerted by the cell wall over the protoplast is called wall pressure. Next, normally wall pressure is equal and opposite to target pressure. Target pressure keeps the soft organs like flowers, young shoots, leaves in stretched form. It is also essential for the growth of cells. Opening and closing of stomata are caused by changes in their target pressure. When a cell is placed in a hypertonic solution, water begins to pass out of the cell by osmosis with a consequent shrinkage of protoplasm. This phenomena is called plasmolysis. When a plasmolysed cell is placed in a hypertonic solution or pure water, endosmosis takes place, as a result of which water is reabsorbed by the protoplast of the cell and the cell regains its original position. This phenomena is known as deplasmolysis. Plasmolysis is of great significance because it helps in determining the living nature of the cell. It is of great help in preservation of meat and fish by salting. It is helpful. Plasmolysis is helpful in preservation of jellies and jams by adding concentrated sugar solution to them. It helps in the plasmolysis of spores of bacteria and fungi. Roots absorb water near their tips in the region of root hair. Root hair have a large surface area. These are tubular outgrowths of epiblema cells. Each root hair has a thin semi-permeable cell wall, a semi-permeable protoplast, and an osmotically active sap present in central vacuole. Mineral absorption is an independent process as minerals are not absorbed with water. It is of two types, passive absorption and active absorption. The passive absorption of mineral takes place along the concentration gradient while active absorption takes place against the concentration gradient. Root pressure is the pressure which is developed by the cortical cells of the root by which liquid contents are forced into xylem of the root and onwards into the xylem of the stem. To be precise, root pressure is the pressure developed by the roots. Root pressure is governed by external factors which influence absorption of water. The upward movement of water from root to the top of the plant is called ascent of sap. Movement of water inside the plant is called translocation or conduction of sap, but most of it is in upward direction, so it is generally called ascent of sap. উপরে ওঠে বলে it takes place through tracheids and মানে ascent of sap takes place through tracheids and vessels of xylem tissue. Next, there are several forces which contribute to the upward movement, but two are most important. Root pressure root pressure builds up sufficient force to push the sap in xylem vessels up to certain height and may be enough for herbaceous plants. Capillary of xylem vessel causes water from a lower level to rise up to fill up the vacuum created by the loss of water due to transpiration from the leaves. The pulling force provided by the leaves is especially important in all trees which do not have enough root pressure. This upward pulling force may also be called as transpiration pull. Downward movement of sap is relatively simple. The food manufactured in the leaves is absorbed in water and it flows down mainly on account of force of gravity. Next, name the following. The process in which water molecules diffuse from region of higher concentration to a region of lower concentration through plasma membrane osmosis. The term given to the osmosis in which water from living cells diffuse out, exosmosis. Shrinkage of cytoplasm towards the center of the cell if it is placed in a concentrated solution of sugar. Plasmolysis, the process of movement of the molecules of a substance from a region of higher concentration to a region of lower concentration. Diffusion, the upward movement of water and minerals from roots to leaves and other parts of the plant. Ascent of sap, the pressure that can be developed in a solution when the solution is separated from a pure solvent by a semi permeable membrane. Osmotic pressure, the pressure that is exerted on the cell wall by fluid contents of the cell. Target pressure, reverse of plasmolysis, deplasmolysis, the tissue which conducts manufactured food from leaves to other parts of the plant. Phloem, a solution whose concentration is lower than that of cell sap. hypotonic solution. Next, fill in the blanks with suitable words. Absorption of water in plants take place through the roots by the process of osmosis. Osmosis is the diffusion of water across a dash membrane, semi-permeable membrane. The upward movement of water along with the mineral salts dissolved in it is called ascent of sap. The pressure by which cortical cells of the root force water into the xylem is called root pressure. In tall trees, ascent of sap takes place by transpiration pool. The diffusion of a dash across a semi-permeable membrane is known as osmosis. The diffusion of a solvent across a semi-permeable membrane is known as osmosis. Dash is the state of cell in which the cell wall is rigid and stretched by the increase in volume of vacuole by absorption of water. Turgidity. The condition opposite to turgid is called flaccid. Absorption of water by the root is by means of root hairs. Water rises in the stem through xylem. Amount of solutes and external pressure affect water potential. True. The true false. Amount of solutes and external pressure affect water potential. True. Turgidity or flaccidity is because of water contents of the cell. True. All plant membranes are semi-permeable in nature. False. Movement of water in vertical direction is called ascent of sap. True. Endosmosis is not observed in all cells. False. Root hair is thin, delicate prolongation of cortex. False. Cell wall acts as a permeable membrane. True. When a cell is placed in a solution of higher concentration, water diffuses out of cell. True. The rate of diffusion of gases is faster than solids or liquids. True. That root hair is an extension of root cells. True. The solution which has the same concentration of dissolved particles as inside the cell is called isotonic. In tall trees, the ascent of sap takes place by transpiration pool. Absorption of water by roots is by means of root hairs. The diffusion of particles of solvent across the semi-permeable membrane is known as osmosis. Huh? The diffusion of particles of solvent across the semi-permeable membrane is known as osmosis. The pressure by which the cortical cells of the root force water into xylem is called root pressure. Shrinkage of protoplasm is called plasmolysis. Due to turgidity, guard cell becomes dash more convex. The plasma lemma of root hair is dash. Plasma lemma of root hair is semi-permeable. The root cap acts as a protective tissue. The loss of water in the form of droplets through leaves and intact plant is termed as guttation. Differentiate between the following pairs: osmosis and diffusion. Diffusion and osmosis. Osmosis or diffusion. Osmosis is the process in which water molecules move through a semi-permeable membrane from a region of higher concentration to a region of lower concentration. Diffusion. Diffusion is the movement of molecules of substance, মানে solute, solvent, gas, সবকিছুই liquid, solid, from a region of higher concentration to a region of lower concentration. এবার osmotic pressure or turgor pressure. Osmotic pressure, the pressure by which the water molecules tend to cross the semi-permeable membrane is called osmotic pressure. Turgor pressure. In a turgid cell, the pressure of cell contents on the cell wall is called turgor pressure. Next question, plasmolysis or deplasmolysis difference. When a cell is placed in a hypertonic solution, water begins to pass out of the cell by osmosis with a constant shrinkage of protoplasm. This phenomena is called plasmolysis. Deplasmolysis. When a plasmolized cell is placed in a hypotonic solution of pure water, endosmosis takes place, as a result of which water is reabsorbed by the protoplast of the cell and it regains its original position. This phenomena is known as deplasmolysis. A turgid flaccid difference. Turgid. When a cell reaches a state when it cannot accommodate any more water, it is fully distended. It is called turgid. Flaccid. The cell in the state of plasmolysis is said to be flaccid. Endosmosis, exosmosis difference. The increased diffusion of solvent across a semi-permeable membrane is called endosmosis. It occurs when a cell is placed in hypotonic solution. Exosmosis. The outward diffusion of a solvent across a semi-permeable membrane is called exosmosis. It occurs when a cell is placed in hypertonic solution. Select the odd one out. Impermeable, permeable, semi-permeable, selective permeable, selectively permeable, impermeable. Plasmolysis, exosmosis, endosmosis, turgidity. Osmosis, ascent of sap, diffusion, imbibition. Hypotonic solution, turgid, endosmosis, flaccid. আর যা নাম্বার 5 নেই। Oh, number 5. Soil, water, xylem, osmosis, root hair. Xylem. What is osmosis? Osmosis is the process in which water molecules diffuse from a region of higher concentration to a region of lower concentration through a semi-permeable membrane. What is exosmosis? When the cell is placed in a concentrated solution, the cytoplasm shrinks. It is due to the diffusion of water from cell to outside, and this process is known as exosmosis. What is endosmosis? Endosmosis is the process of osmosis in which water enters in living cells from hypotonic solution. What is turgor pressure? The upward movement of water and minerals from root to leaves and other parts of the plant is termed as turgor pressure. What is turgidity and flaccidity? A fully distended cell due to absorption of water is called turgid cell, and this condition is described as turgidity. A cell which is no more turgid is called flaccid, and the condition is described as flaccidity. What is total plasmolysis? When water from cell is removed completely out of the cell due to exosmosis, the protoplasm shrinks to the maximum. The condition of the cell in which no more shrinkage is possible is known as total plasmolysis, under which osmotic condition water does not enter into cell. When target pressure becomes equal to wall pressure. When both internal and external solutions reach equal concentration, then further entry of water into the cell does not occur. Name two substances which enter the root through root hair. Water are soluble mineral ions from the soil. Give one example of the osmosis occurring in human body and in green leaf. In human body, movement of water into blood plasma form the bloody fluid. Movement in a green leaf কি হয়? Movement of water from leaf veins into the mesophyll cells. What is wall pressure? When protoplasm is pressed against the cell wall due to target pressure, the cell wall also exerts an equal and opposite pressure. The pressure which is developed from the cell wall is called wall pressure. What is flaccid condition of the cell? When cell is plasmolized, it loses its turgidity. Due to this, the cell loses its shape and shrinks. Such a shrunken condition of cell is said to be flaccid. Define ascent of sap. The process by which water absorbed by root is carried to the aerial parts of the plant is called translocation of water or ascent of sap. Define diffusion. Diffusion is defined as the movement of solutes through the solvent from their higher concentration to a lower concentration. Gas is also diffused due to the random movement of molecules. What is the importance of diffusion? Exchange of gases during photosynthesis and respiration is a diffusion process. Osmosis is the diffusion process where only solvent is allowed to diffuse by preventing the movement of solute particles. It spreads molecules and ions throughout the protoplast. It keeps plant cell wall moist. Next, can osmosis be regarded as diffusion? Give reason. Yes. Water diffuses from a weaker solution to a stronger solution per unit volume. So it is a kind of diffusion occurring through a semi-permeable membrane. What is root pressure? The force causing the flow of sap from lower level of the stem of a plant to a higher level is known as root pressure. Where bacteria fungi do not grow in pickle, jam and jelly. Why? Jam, jelly, and pickle contain higher amount of sugar or salt. This makes them hypertonic, in which plasmolysis of bacteria and fungi takes place. This kills the bacteria and fungi, and hence pickles, jams, and jellies are preserved properly. Why is it better to transplant seedlings in the evening? It is better to transplant seedlings in the evening because in the evening the stomata is closed, so there will be no loss of water from the plant body and it will get sufficient time to fix its roots in the soil. But in the morning it will lose water by transpiration and may die due to the lack of water. Differentiate between plasmolysis and deplasmolysis. Plasmolysis takes place when a plant cell is kept in a hypertonic solution. The protoplasm of the cell shrinks away from the cell wall. Plasmolysis results in the flaccid condition of the cell and the plant. Deplasmolysis, it takes place when plasmolysed cell is kept in hypotonic solution. Deplasmolysis, the protoplasm of the cell swells up and touches the cell wall. Deplasmolysis results in the turgid condition of the cell and the plant. Mention some importance of water. It is a universal solvent. Water is a medium of transport for all kinds of materials. Water is useful as a lubricant and helps in transport of gases like oxygen, carbon dioxide during respiration. Water helps in the maintenance of turgidity of cells. Explain why the raisins swell up in water. The dried grapes from raisins, when they are kept in water which is hypotonic, it results in endosmosis that swells up. Which part of root hair acts as semi-permeable membrane? Cell membrane or plasma lemma? What is incipient plasmolysis? The initial stage of plasmolysis during which protoplasm sinks away from the cell wall is known as incipient plasmolysis. Differentiate between hypotonic and hypertonic solution. In hypotonic solution, the solute concentration is lower than that of cell sap, but in hypertonic solution, the solute concentration is higher than that of cell sap. What is osmotic gradient? Osmotic gradient is the difference in concentration established between two solutions across the cortical cells. What is imbibition? It is the phenomena by which living or dead cells containing colloidal particles absorb water by surface attraction. What are the main characteristics of diffusion? Main characteristics of diffusion are: diffusion always takes place from a region of higher concentration to a region of lower concentration. The rate of diffusion depends upon the size, temperature, and contraction of its molecules. Rate of direction of diffusion molecules are independent by the molecules of other substances present. What is semi-permeable membrane? It is a membrane which allows the solvent to pass through it but not the solute. What is isotonic solution? An external solution having the same solute concentration as that of cell sap is known as isotonic solution. What are the factors which influence turbidity of cells? Adequate supply of water, semi-permeable membrane, presence of osmotically active substances in the cell. What is the significance of osmosis? Osmosis regulates the opening and closing of stomata in plants. Osmosis maintains osmotic balance by osmoregulation in animal cells. Osmosis plays an important role in the absorption of water by plants from the soil. What is suction pressure? The net force with which the water is drawn into the cell is called suction pressure. What is a solvent? A liquid in which another substance is dissolved to form a solution is called solvent. What is capillary? When a narrow tube is placed in a beaker containing water, the level of water rises in the tube. The phenomena is known as capillary. Capillarity. নেক্সট ছবি আছে একটা ইউ এর মতো দেখতে, তার ভেতর সুক্রোজ মলিকিউলস আছে, আর 5% সুক্রোজ সলিউশন, ওয়াটার মলিকিউলস 10% সুক্রোজ সলিউশন, এইসব। সেমি পারমিয়েবল মেমব্রেন আছে, সেমি পারমিয়েবল মেমব্রেন দিয়ে 10% সুক্রোজ সলিউশন থেকে 5% সুক্রোজ সলিউশন এ থেকে বিতে হয়ে গেল। হুইচ ফেনোমেনা ইজ ইন্টেন্ডেড টু বি শোন বাই দ্য রেস্ট এক্সপেরিমেন্ট? অসমোসিস। হুইচ লিম্ব অফ ইউ টিউব contains more concentrated sucrose? Limb A of U tube contains more concentrated sugar solution. Why have the two kinds of molecules been shown in different sizes? To represent solute and solvent molecules. Why is the membrane separating the two solutions labeled as semi-permeable membrane? Semi-permeable membrane allows two solvent or water to go through it. It does not permit the solute particles across it. Which limb of U tube is functionally compared to the root hair of a plant? Limb A of U tube is functionally compared to the root hair of the plant. নেক্সট দি অ্যাপারেটাস গিভেন বিলো একটা ওই জল এরকম গোটস ব্লাডার বলছে ওটাকে, ওই এটা দিয়ে আটকানো আছে জল, থিসেল ফানেল স্ট্যান্ড বিকার পিওর ওয়াটার সুগার সলিউশন এরকম গোটস ব্লাডার বলে একটা মানে একটা ছবি আছে এটা। what process does the experiment demonstrate? Osmosis. Name two liquids that could be used in the experiment. Distilled water or concentrated sugar solution এই দুটো লিকুইড would be used in the experiment. Why did the level of liquid in the vertical tube rise? To rise in the level of vertical tube can only be due to the entry of water into it through goat's bladder by means of process of osmosis. Explain how the process named in one affects a protozoa such as amoeba. In amoeba, osmosis helps in maintaining water balance. Give one example in plants in which the above process occurs. What is the advantage of this to the plant? Root hair absorbs water from soil by osmosis. একটা ছবি আছে, if the membrane is to behave as a semi permeable membrane, what can be stated about the size of pores? Pores must be smaller than the solute molecules but larger than solute molecules. Will solvent molecules pass through the membrane from left to right, from right to left, or in either directions? Solvent molecules will pass the membrane in both directions. Since solvent molecules are present on both sides, they will strike the semi permeable membrane and pass through the same. By which direction will there be a net movement of solute molecules? There is a net movement of solute molecules from a region of a higher chemical potential to a region of lower chemical potential from left to right. Name the apparatus arranged in the given figure signifies an important process. Name the process. Osmosis. Where does the process occur in plants? Root hair. What solution is placed inside the dialysis tubing? Sugar solution. What happens to the level of the solution in the capillary tube? The level of the liquid in the capillary tube rises. What happens? Does the process define the process mentioned in question 1? Osmosis can be defined as the process of movement of solvent from the region of higher concentration to a region of lower concentration through a semi-permeable membrane. এবার একটা ডায়াগ্রাম দিয়েছে, what is the state of cell shown in the diagram? Flaccid condition. Then the structure which acts as a selectively permeable membrane, plasma membrane. If the cell has been placed in a distilled water instead of strong sugar solution, which feature would not have been present? The sunken protoplast would not have been present. If the cell in the diagram processed chloroplast, where would there be present? The chloroplast would be present in the sunken protoplast. Name one feature of this plant cell which is not present in animal cell, the presence of cell wall. এবার একটা ছবি আছে, নেম দ্য পার্টস ইন্ডিকেটেড বাই দ্য গাইডলাইন্স 1 to 4. Vacuole, solid particles, xylem vessel, cortex cell. Is the root hair unicellular or multicellular? Mm, unicellular. Name the process responsible for the entry of water molecules from the soil into A1 and A2. Osmosis. The pressure is responsible for the movement of water in the direction indicated by arrows. Osmotic pressure. How is the pressure set up? This pressure is set up due to the difference in osmotic gradient.